speakings of the day. I am Dr. Shweta Bavari from Pharmacy Institute, Noida Institute of Engineering and Technology, Greater Noida. In this lecture, I am going to cover the topic pathophysiology of infectious diseases, meningitis and typhoid basically. Beginning with the pathophysiology of infectious diseases. Infectious disease can be defined as an illness due to a pathogen or its toxic product. So, for example, the illness can be due to Clostridium tetani or the tetanus toxoid or tetanospasmin that it produces. So, infectious diseases are basically the disease that are caused due to a pathogen, infectious microorganism and the products they produce. So, infectious diseases are caused by infectious organisms, typically bacteria, viruses, fungi or helminths. And apart from these, there can be other ectoparasites, there can be protozoans also that cause infectious diseases. It arises through transmission from an infected person, an infected animal, or a contaminated inanimate object to a susceptible host. Inanimate object means non-living objects. So from there also they can transfer, like the utensils, they can be clothes, like handkerchief which is contaminated with the bacteria or these microorganisms that can cause infection. So this is generally how they transmit. In this lecture, basically, we are going to focus on two major types of infectious diseases, meningitis and typhoid. So we'll first begin with the pathophysiology of meningitis. As you can see, meningitis, it is made up of two words. One is meninges, the second one is itis. Meninges means the protective membranous coverings of the brain and spinal cord. If this will help you recognize, you would have you have already studied this in HAP, that meninges include the outermost dura mater, the middle one arachnoid mater and the innermost pia mater. These are the protective coverings of the brain and the spinal cord. And we are talking about meningitis. Itis stands for inflammation. So any infection, we are here focusing on the infectious disease that can cause inflammation of these meninges. So meningitis is defined as inflammation of the membranes of the brain or spinal cord, specifically the subarachnoid space. In the figure, you can focus the subarachnoid space is the space, the cerebrospinal fluid, CSF filled space between the pia mater and the arachnoid mater, that is known as subarachnoid space. So basically the infection lodges in this particular portion and results in the inflammation of these protective membranes or the linings of the brain and spinal cord, that is meningitis. Meningitis occurs following an infection localized to the subarachnoid space that I just told. The symptoms of meningitis range from flu-like symptoms, which include fever, chills, muscle aches, cough, congestion, runny nose, with severe headache and high fever. It may result in a stiff or painful neck, then altered mental status. There can be confusion, there can be drowsiness, there can be convulsions, there can be irritation due to bright light. It may result in joint pain, cold hands and feet, vomiting. The cranial nerve defects are seen in 14 to 33% of cases and result from inflammation surrounding cranial nerves that traverse the subarachnoid space. We talk about cranial nerves, we talk about the 12 cranial nerves, 12 pairs of cranial nerves that we have already studied in human anatomy and physiology. So we talk about cranial nerves. The two of the cranial nerves have basically sensory function, and apart from those, the other 10 cranial nerves have mixed functions, sensory as well as motor. So when there is a infection and inflammation in the subarachnoid space, the nerves that are supplying, the nerves that innervate that particular region, the subarachnoid space, they also become affected, and this results in lot of alterations in the sensory functions and the motor functions of these cranial nerves. Main causes of meningitis. What are the causative agents for meningitis? Meningitis is basically caused by bacteria, viruses, fungi. Then we talk about bacterial meningitis. The most common causative agents of bacterial meningitis are Streptococcus pneumoniae, Haemophilus influenzae, Neisseria meningitidis, Listeria monocytogenes, and Staphylococcus aureus. So these are the bacteria that can lodge infection into the subarachnoid space and cause inflammation of the meninges, resulting in meningitis. Now we talk about the viral meningitis. So the most common causative agents of viral meningitis are enteroviruses such as Coxsackie and ecovirus, mumps virus, herpes virus, and human immunodeficiency virus. Fungal meningitis, the most common causative agent of fungal meningitis is Cryptococcus neoformans. Bacterial meningitis, bacterial meningitis results in an acute form of meningitis, which is a medical emergency. It is an important cause of morbidity and mortality if left untreated. If in that medical emergency situation, if rehabilitation is not provided or the medical treatment is not provided on urgent basis, it may even lead to death. So this is bacterial meningitis is the most severe forms of meningitis as compared to viral meningitis and fungal meningitis. It results in acute infection, as I said. Bacteria, after colonizing nasopharynx, eventually reach the CNS. Generally, the bacterial infections in the nasopharyngeal region, the nasal root and the pharynx, where the bacteria generally house, resulting in respiratory infections. From after causing respiratory infections also, these bacteria traverse their path. They pave their path to the subarachnoid space, where they lodge another infection, resulting in meningitis. Viral meningitis is more common as compared to bacterial meningitis and fungal meningitis. Viral meningitis is more common, but it is comparatively less serious form as compared to the bacterial meningitis, because bacterial meningitis can result in mortality and morbidity. It is fatal. It is a medical emergency, causes acute form of infection that requires immediate medical attention. But viral meningitis, they are more common as compared to bacterial meningitis, but they are less severe forms. Most episodes resolve spontaneously and rarely result in death. Fungal meningitis, this is rarest of all. This type of meningitis is most commonly seen in people with HIV infection, or you can say in immunocompromised people, in people like, for example, who are undergoing cancer chemotherapy. So those diseases, those type of infections, those immunocompromised states which allow opportunistic infections to occur, in those we can find fungal meningitis, which is rarest of all. It is not of common occurrence. Then we talk about pathophysiology of meningitis, how this infection occurs in human host and how the disease progresses, that we'll talk about. Meningitis begins with invasion of subarachnoid space by pathogens, mostly through bloodstream to brain, from other sites of infection in the body, and rarely through skull fractures, surgeries, etc. So the invasion by these pathogens, these bacteria, virus, fungi, this occurs, the invasion into the subarachnoid space occurs usually from other sites of infection. As I already told, that bacteria that have already caused a respiratory infection, from there they can pave their path to the CNS, they can lodge an infection in the subarachnoid space. Similarly, from other sites of infection also, the sites can be systemic infection, the infection that is that have reached the blood. So any other site of infection, from there also these pathogens can pave their path to the meninges, okay, and then they can lodge infection. They can invade the subarachnoid space, the space between the pia mater and arachnoid mater, which is filled with CSF. So rarely this occurs that if there are open wounds in the head, there was an accidental case, there can be cases of surgery, then there can be skull fractures. In those cases, there can be open wounds. So from directly from there, the infection can lodge into the subarachnoid space as well, but this is rare. This rarely happens. The systemic inflammatory response of host, leads to leukocyte extravasation into subarachnoid space and increased CSF outflow resistance and brain edema. Now, once an infection has lodged into the subarachnoid space, there is infection. So, when there is an infection, there is going to be triggering of the host immune response. So, host immune response is triggered by these pathogens that invade the subarachnoid space, that invade the meninges. Okay, and due to the triggered immune response, there is infiltration. Lot of leukocytes from they come to the site of infection, they reach the subarachnoid space. So lot of neutrophils, lot of leukocytes. When I talk about leukocytes, there are granulocytes and agranulocytes. So I'm saying lot of leukocytes enter the site of infection. This results in so when there are lot number of leukocytes, there is some sort of congestion in the subarachnoid space, so that the CSF cannot drain out of the subarachnoid space. There is restriction, there is resistance to the outflow, the flow of the cerebrospinal fluid from that subarachnoid space. CSF accumulates there, it cannot go out. So this accumulation of the cerebrospinal fluid results in brain edema. Okay, it results in brain edema. Now, when there is brain edema, there is so much accumulation of the cerebrospinal fluid. This results in elevated intracranial pressure. The intracranial pressure is elevated, it increases, and when the pressure is increases, this results in compression of the brain tissue against the skull. The brain tissue is compressed, it is pressed against the skull, which results in ultimately the inflammation of the meninges and the brain tissue, as well as death of the brain cells. Also, immune cells within the brain, like microglia, are stimulated by pro-inflammatory microbial products, which can directly lead to neuronal injury. Now, because of these pathogenic organisms, these microorganisms, the reticuloendothelial system in the brain, in the form of microglia. What are microglia? They are basically monocyte macrophage system present in the brain itself for defense, for the purpose of defense. So they are triggered, and they basically result in what? A pro-inflammatory response. Pro-inflammatory mediators are released into in the near vicinity of the brain, and these mediators, although their target is the microorganism, still they have additional effects. They also target the neurons. They result in neuronal injury also. They result in destruction of neurons also. Neuronal injury is also mediated by the release of proteases, cytokines, and excitatory amino acids. And other than this microglia, this neuronal injury also occurs due to the release of proteases, the enzymes that cause destruction of the proteins. So the proteins are also present in the neurons, the cells, the brain cells of the host as well. So they also get destructed along with the microorganisms. Then cytokines like interleukin 6, interleukin or tumor necrosis factor alpha. So they result in the necrosis or damage to the neurons. Apart from that, excitatory amino acids like glutamic acid, they also result in neuronal damage, neuronal injury. This results in meningeal inflammation, which may exacerbate to coma and even death, because there is extensive neuronal damage, death of the brain cells that can eventually lead to coma, and it can also result in death. The mode of transmission of the causative agents of meningitis. So the causative agents of meningitis, whether be bacteria, viruses or fungi, they spread from person to person through close contact with respiratory secretions or saliva, such as coughing, sneezing and kissing. Enteroviral meningitis spreads through fecal oral route. When we talk about fecal oral route, we are talking about contaminated food, improperly cooked food, the uncooked food, and contaminated water consumption. Sharing eating utensils, glasses, food, or even same room can also spread these microorganisms that cause meningitis. Moving ahead to the pathophysiology of typhoid. Typhoid fever, we have already heard about typhoid a lot, and it is a very common infection. Typhoid fever is an enteric fever characterized by systemic illness along with severe abdominal pain and fever. So when we talk about typhoid fever, first of all, we are talking about enteric fever. the infection which basically focuses, or the infection that occurs in the gut, basically in the intestinal region, and it is characterized by systemic illness. Systemic illness means this infection spreads to the systemic circulation, to the blood, and it is basically characterized, the symptoms basically include abdominal pain and fever. These are the characteristic features of typhoid fever. The causative organism of typhoid is Salmonella typhimurium, which is a gram-negative anaerobe. When I use the term gram-negative anaerobe, gram-negative bacteria are the ones who have very thin peptidoglycan cell wall, first of all. Anaerobes are microorganisms that can thrive in oxygen deprivation also, in the lack of oxygen also. Mode of transmission of Salmonella typhimurium. Salmonella is transmitted by the fecal oral route through contaminated water, undercooked food, and fomites. Now, I already told fecal oral route is that is the consumption of contaminated water and or uncooked food or contaminated food. Okay, and fomites are belongings of the infected person, such as using their clothes, using their utensils, sharing food with them, and using their furniture, using their bed sheets, using their handkerchief. So this can also transmit the infection from person to person. It is more common in areas with overcrowding and poor sanitation. It is only transmitted from an infected person to another person, as humans are its only host. Salmonella typhimurium do not have any animal host or do not have any other vector who can transmit this infection to the human beings. All right, it transmits from person to person, from human to human only. There is no animal vector in between. Okay, because humans are the only host for Salmonella typhimurium. Now we discuss how this infection occurs in human beings and what are the stages of disease progression of typhoid. Although Salmonella typhi is an acid-sensitive bacteria, but when enters into the gut through oral route, its colonization in human body becomes easy if a person is consuming highly contaminated food, consuming drugs like antacids or anti-ulcer drugs, or even food and beverages that have buffering effect against gastric acid. Now, what does this mean? It states that Salmonella typhi or Salmonella typhimurium is basically what it is acid. It is acid sensitive. The gastric acid secretion in our stomach, the acid in our stomach has the ability to kill this bacteria. But still, this is a very common form of infection. How does this occur? This can occur if a person consumes highly contaminated water or food. The food which is containing the large concentration of this Salmonella typhimurium. The food is highly contaminated or water is highly contaminated with this bacteria. Or in cases where the person is consuming drugs like anti-ulcer drugs, antacids that have ability to reduce acid secretion. So acid cannot, when the acid secretions are low, it cannot destroy Salmonella typhimurium. Then there can be the dietary habits of individual also impact the same. Like a person who resorts to eating more junk food, that have the food that has a capability to reduce acid secretions, or consuming beverages that have the ability to reduce acid secretions. In those people also, this Salmonella typhi can easily enter through the oral route. The virulence of Salmonella, the severity of the infection by Salmonella, is determined by typhoid toxin, VI antigen, which is VI antigen is basically what? Polysaccharide capsule, the polysaccharide capsule of the bacteria, this Salmonella typhi. Liposaccharide O antigen and flagellar H antigen. These are the four major antigens. These help the Salmonella typhi to lodge an infection inside a human host. VI antigen acts as an anti-phagocytic agent, preventing the action of macrophages. thus shielding the O antigen from antibodies that confer the serum resistance. So when Salmonella enters the body, first the first antigen, the first virulence factor that comes into play for lodging an infection is VI antigen, the polysaccharide capsule of this bacteria. It comes into play. What is its role? It prevents the phagocytosis of Salmonella typhi by the macrophages of the host body. Okay, so macrophages cannot phagocytose these bacteria because of the VI antigen. So it is resistant, it becomes resistant. Now when macrophages cannot destroy this bacteria, so the O antigen cannot be released. If O antigen cannot be released, so when this bacteria goes into the systemic circulation of the host body or the human body, it goes undetected there also. It doesn't trigger any immune response there also because O antigen is not released. It acts as a shield. So they remain undetected by the host immune system. This is what it is. Then the flagellar H antigen comes into play. The flagellar H antigen, or in simple terms, you talk about just flagella. The flagella of this bacteria provides bacterial mobility and adherence upon the gut wall mucosa. So this is how this bacteria, Salmonella typhi, it travels to the gut. It travels to the gut and then it attaches to the gut mucosa, the intestinal mucosa, using the flagella. Hence, once consumed, typhoid bacteria cross the epithelial layer of the intestinal wall. They are then quickly consumed by macrophages and transported to aggregates of lymphoid tissue in small intestine, where the immune function of the gut is most concentrated. Now when they enter into the gut, they are consumed by the macrophages there and they lodge an infection, but they are able to lodge an infection in the Pierce patches. What are Pierce patches in the intestine, small intestine? Basically, they are lymphoid tissue. Whenever I use the term lymphoid tissues are where B lymphocytes and T lymphocytes are basically concentrated, right? Lot of B lymphocytes and T lymphocytes are present. That is lymphoid tissue, the Pierce patches we talk about. So from small intestine, typhoid bacteria disseminate. It then moves into the systemic circulation, causing a life-threatening systemic illness, typhoid fever, in the first week. So in the first week itself, after lodging an infection in the Pierce patches, from there it paves its path into the blood, into the circulation. As soon as the Salmonella typhi reaches the circulation, there it results in threatening systemic illness. It results in typhoid fever. This occurs in the first week of the infection. The typhoid bacteria then spreads to reticuloendothelial system, resulting in splenomegaly. Now from there, typhoid bacteria again lodges the infection into where? Reticuloendothelial system, the monocyte macrophage system of our body, where it results in splenomegaly. Splenomegaly, it means enlargement of the spleen. Enlargement of the spleen occurs, and liver necrosis, destruction of the liver cells occurs, hepatocytes occurs in the second week of the infection. The typhoid bacteria then colonizes in gallbladder, causing chronic infection. Then ultimately from the blood circulation, it again enters into the gallbladder, and now from here it lodges a chronic infection, and then back to the GIT, ultimately stimulating an immune response that results in ulceration of Pierce patches. So there is an entire cycle from the gut into the Pierce patches, from Pierce patches to the systemic circulation, from systemic circulation from the blood to the gallbladder, and again to the Pierce patches. So this circle keeps on going, and it ultimately results in the ulceration of the Pierce patches. The ulcers form in the Pierce patch, and there are intestinal, the intestinal mucosa is abraded, and there are perforations. Pores are formed in the intestinal mucosa. This results in bleeding from the intestines in the third week of this Salmonella typhi infection. This is how the typhoid disease or typhoid infection progresses. So basically, when we talk about this infection, we talk about first the infection is lodged into the gut, in the intestine. From there, it goes to the Pierce patches. From Pierce patches, it moves into the systemic circulation. From there, it goes into the gallbladder and again to the Pierce patches, and this goes on. And this cycle goes on. And this ultimately results in ulceration in the Pierce patches, ulceration in the intestine, resulting in big holes. When there are big holes, it results in bleeding from the intestine. So this is the pathophysiology of typhoid. The symptoms of typhoid include nausea, vomiting that progresses to diffuse abdominal pain. The entire abdominal region has pain affliction. Then there is bloating, which is gasiness, anorexia, which means aversion towards food, reduced hunger, and diarrhea. Bacteremia means presence of bacteria, Salmonella typhi, in the bloodstream, and fever with flu-like symptoms, that is malaise, pain in the body, the fever and the chills. Fever follows a step-ladder pattern. The fever that occurs in the typhoid fever, typhoid infection, it basically consists of the peaks and the troughs. It will rise suddenly and then it will fall down. Again, it will rise suddenly, then it will fall down. So there is a peaks and trough pattern, which is known as step-ladder pattern. And then ultimately it results in abdominal distress, lot of abdominal discomfort, lot of pain, severe form of pain, and enterocolitis, that is inflammation in the intestinal region. This is all about meningitis and typhoid. Thank you.